All right. Hello, everyone, and welcome to the second episodes of our series highlighting current and exciting research projects in the MGNet Research Consortium. My name is Anusha, and I am a program manager at MGNet, and I will be your host for today. I am joined with Dr. Kevin Lee, researcher in the Department of Neurology at Duke University School of Medicine. He was selected as the 2021 MGNet Scholar. I am also joined by Mr. Rondell Jones, our MGNet patient advocate. In this episode, we will feature Dr. Lee's project titled Investigation of CD8 T cells immune signatures in myasthenia gravis. I now will give you both the floor to discuss this important topic in MG research.
Hello, Dr. Lee.
Hey, Ms. Jones, how are you today?
I'm doing well, thank you. First and foremost, congratulations on your award, being a scholar.
Thank you.
To get everything started off, can you just tell us what your research is about?
Yeah, sure. Thank you for the question. So before I respond your question, I'd like to express my sincere gratitude to the organizer for this invitation. So it's really a great honor for me to be part of the MGNet Scholar program. So I would also like to extend my thanks to MGNet, the MGFA, and especially to the patients with myasthenia gravis who participate in this study. So for over a decade, my journey with myasthenia gravis research has taken me from China, where I was deeply involved in clinical trials and translational researches, and to the United States to advance my career and pursue innovative research in this field. So the core of my work focuses on the understanding the mechanisms underlying myasthenia gravis and identifying potential biomarkers in MG patients. So as you know, myasthenia gravis is known to be an autoimmune disorder characterized by the production of autoantibodies that impair the neuromuscular junction, leading to muscle weakness and fatigue. And this involves a very complex immune responses. So my overall research has primarily focused on to understanding the role of immune cells in developing of MG and try to find the useful biomarkers. So this project concentrated on a subset of immune cells known as CD8 T cell within the context of myasthenia gravis. So we will observe that the CD8 T cell in patients exhibit a significant heightened status of activation compared to those in health controls. Interestingly, we found that the levels of these hyperactivated CD8 T cell declined following treatment with azathioprine, so indicating its efficacy in suppressing this specific subset of CD8 T cells. In addition, we identified a subpopulation of CD8 T cells that turn exhaustion, characterized by prolonged exposure to an antigen, which result in T cells with distinct surface markers and a reduced response to stimulation. So in MG patients, we observed an elevated count of exhausted CD8 T cells, so which notably decreasing following treatment with immunosuppressant. So overall, our research collectively underscored that both hyperactivated and exhausted CD8 T cell population are increased in MG patients, and significantly, we have identified azathioprine as an effect agent inhibiting these specific subsets of CD8 T cells. Thank you.
Okay. Thank you for telling us a little bit more about your research. So, why is it important what you found with the cells with regards to myasthenia gravis patients?
Yeah. So this is very important because in the clinic, at the moment, the treatment for MG is somewhat of a one-size-fits-all approach. So it can be a process of trial and error to find the right treatment for each individual patients. But by identifying specific biomarkers, we aimed to personalize treatment plans. This means we could potentially start with the most effective treatment for MG patients right away, rather than trying several different medications. So additionally, by understanding the behavior of these immune cells, we may be able to predict the causes of the disease, helping us to treat it more efficiently. So this is why biomarker research is so important, and continuous supporting biomarker researchers area is so critical, especially from the MG patient community.
So with that, what do you think is the most important insight you can give me as a patient, and how will it impact me as well as others with myasthenia gravis?
Oh, yeah. Sure. So one of the key insights for our research is that the levels of this exhausted and pro-inflammatory of CD8 T cells can give us clues about the severity of MG in a patient and how they might respond to treatment. So for patient, this could mean a more streamlined path to find the most effective treatment with fewer side effects, less wasted time, and better overall of management of conditions. So it also helps us understand the disease better, so we can start to develop new treatments that specifically target on those immune cells subsets.
Right. That sounds great. A vast improvement, I guess, over the years. So, do you feel research benefits newly diagnosed patients or more established patients or long-term patients like myself? I've been diagnosed for 30 years with myasthenia gravis. So, do you feel it sways either way to newly diagnosed or veterans, so to speak?
Yeah, so the biomarker research, so does benefit either newly diagnosed MG patients or long-term MG patients as well. So as we discussed above, so we aimed to personalize treatment plans. This means we could potentially start with the most effective treatment for newly diagnosed MG patients, or we could also switch to a new effective treatment if the current treatment waning off in the long-term MG patients. So our final goal is that the biomarkers will also be available to monitor the immune system changes with clinical symptom fluctuation. So this ultimate goal will benefit the overall MG community.
Wow, that's great. Just sounds like, again, research is, and there's more improvement with research. So with that, what is the future of your research, short term and long term? And you can determine what you consider short term and long term.
Yeah, so the future of our research is geared towards confirming these biomarkers in a very large group of MG population. So we'll further explore biomarkers between the treatment response group and treatment non-response group. And we are also looking at whether these biomarkers can give us a real-time insight into how treatments are working. So in the long run, we hope that this research will lead to a test that could be used in the clinic to guide the treatment decisions. So beyond that, we are excited about the possibilities that our findings could lead to the development of new drugs that target this on these specific T cells, which could be more effective and have fewer side effects than the current treatment. And in the near future plan, so as I discussed previously, so we are focused on the biomarkers between the treatment response group and the treatment non-response group, so we can find we will identify the more useful biomarkers between them. So that will be our near future plan for our research.
Good, good. I just want to say thank you for your time, Dr. Lee. It was a pleasure talking to you and hearing more about your research. Is there anything else you would like to share with the MGNet community in closing?
You're so welcome, and thank you so much for your time today. And again, I want to thank the supporters from MGNet, MGFA, and the MG patient community, and looking forward to contributing more science to MG research. Thank you.
Thank you.
Thank you both so much for participating in this conversation. I think these discussions are super, super important for patients and their families. It's super helpful. We hope that this conversation will be a great resource for the MG community, and I thank you both for your time.
Thank you.
Thank you.